Okay, so this week, bringing you a lecture about principal component analysis. As a reminder, you have a reading due or summary for your reading due this Friday at midnight. Canvas is set up for midnight Mountain Standard Time, and unfortunately, I can't change that. So if you are somewhere not in Colorado, make sure that you are aware of that. And if it's a problem or you missed it or something like that, just email me. Okay, so the outline for lecture today, we're going to go over what principal component analysis is and why we might want to use it. We're going to briefly go over how to calculate a PCA. You're not going to be asked to do this by hand, mostly because now like R does it for you. But I do think it's useful to understand sort of some of the basic steps and basic components of the calculation, mostly because PCA is part of a larger suite of statistical tools called multi-dimensional scaling techniques. And so there are some nuances between the different techniques, and I think it's useful to understand sort of how PCA works so you can determine if it's the most appropriate statistical test to use. More importantly, we're going to go over how to interpret the PCA results and then conclude with briefly going over the assigned paper. So PCA is a technique for reducing the dimensionality of a data set or interpreting multiple variables in a two-dimensional space, which is a little bit wordy, but it's not as complicated as it sounds, and it uses mostly math that you're already familiar with. But to illustrate the use of a PCA and sort of what this reducing dimensionality means, we're going to kind of walk through a pretty common application of a PCA, which is analyzing the genetic makeup of microbial communities. So a little background on the illustrative example. My research focuses largely on the seasonal changes in nitrogen biogeochemistry in tropical lakes. And one way that we look at that is by looking at microbial communities. So microbial communities in both the sediment and the water column are responsible for a lot of the biogeochemical pathways that take place in a lake. And so we can infer the microbial function based on the genes in a microbial community. So we won't get into it because this isn't a genetics course, so don't get caught up in those details. But if you're into microbes and genetics, we can talk about that another time. So in doing this sort of analysis work, we can't measure every single microbe in a sample and identify it to the species level or even like the genus level, because that would be computationally, logistically, and financially impossible. However, we can measure the genes of the whole sample and start grouping similar communities together. So imagine you have a landscape or a lake, sort of any, pick your favorite ecosystem, and you take samples from multiple different locations. You can analyze the genetic components of those different samples and start looking at which locations, which samples are most similar. And sort of the second level is like, what are those genes? What functions do they infer? But if the microbe part is throwing you off, think about your own genes, right? Your genes would be most similar to your siblings. If you have those, I don't, but I hear they're great. Then your biological parents, then your grandparents, cousins, and so on. So the more genes you share, the more similar you are in sort of a genetic landscape. So here's our example. We have 7 different samples. So you can think of it as 7 different locations or 7 different water samples. And then 6 different genes just called genes A through F for simplicity. And for the purposes of the example, I just use sort of a unitless number. So you can think of 0 as being none of that gene present in the sample, and then I think 9 is the highest number used. So 9 being a lot, that gene being really abundant in a certain sample. So you have these 7 different locations, 7 different samples, and 6 genes. And you want to know which one of these samples, which one of these locations are most similar. So if you look at, and again, if the microbe gene part is throwing you off, samples could be states, and variables could be components of the economy, so like GDP or something. Similarly, samples could be high school students, and the variables could be test scores in different classes, like your math final, English final, whatever. Back to microbes. So if we just want to look at the first gene or the first variable, you could kind of put the samples on a traditional number line. Based just on gene A, you see that 5 and 6 are most similar, 2 and 1 are most similar, and then 4, 7, and 3 group together. That's fine, that gives us some information. If we were just interested in gene A, we could stop here. But we're not. We're trying to figure out, using all of the genes available, which samples are still the most similar. So we could look at 2 variables at a time in sort of a traditional XY plot. And looking at just gene A and gene B, we see that, based on sort of the grouping from gene A, we still see that with 5 and 6, 1 and 2, and 4, 7, and 3. And gene B doesn't really add a whole lot of information. It doesn't further separate out any groups. So it'd be tempting to stop here and say that, that's how they group together, but there's still 4 more genes that we haven't accounted for. So trying to add a 3rd gene, we could look at a 3-dimensional space, right? Because each gene is on its own axes. But then you get something that starts to look like this, and it's really hard to wrap your brain around more than 3 dimensions. Even I think looking at just the 3-dimensional plot is really hard to interpret. If you have more than 3 genes, you would need more than 3 axes. And that's just, unless you're Rain Man, that's not gonna work out for you. So going back to looking at 2 genes at a time, we could work through the data, 1 pair of genes at a time, but it would be really difficult to make sense of. So up top, we have this gene A, gene B comparison again. Then underneath it, we're looking at gene A versus gene C. So where gene B didn't add a whole lot of extra information, gene C separates things out almost too much. We see that samples 3 and 7 still clump together, and everyone else is pretty scattered. So you go through and you create all these pairs and sort of look at it and try to get a sense of, how often your samples clump together. But it would be really hard to do this sort of qualitatively. So what a PCA is going to do is still create these different pairs, but produce one final interpretable final product. So PCAs are used a lot of different ways, but right now we're going to focus on 2 things that a PCA can tell us. Tell us which samples cluster together and which genes or variables are correlated to each other. So the second point, which genes are correlated to each other. You can think of this as in, it's either an investigative tool, right? So in the paper, they found that total phosphorus and total nitrogen were positively correlated, which makes sense if nutrient loading is occurring in a system. Both phosphorus and nitrogen often go up at the same time. Or you can think of it in terms of modeling, right? So models should be as simple as possible, but no simpler. And you'll find that things like evergreen tree cover and NDVI are positively correlated, right? Because trees are green. But in a model, you would use one or the other, but maybe not both. So think about variables being correlated. That's sort of the broader picture. So we do this by calculating covariance and correlations. And this covariance and correlation are sort of the main takeaways that I'm aiming for in this lecture. And this is sort of, a lot of this is the math part that we're going to gloss over. So I'd like you to focus on most of the theory behind it. So we go through this matrix, and you'll recall the XY plots that we said we could create for each pair of genes or variables, right? So just as an example, gene A versus gene B. And within each one of these XY plots, you do a simple regression like you're already familiar with. And note that halves of this matrix will be mirrors, right? So AB, BA. The line will look different, but it's going to have the same correlation, right? And so you can go through this matrix and don't actually calculate it. Those lines are kind of arbitrary for an example. So if you do choose to go back and practice making a PCA with this, your data won't look like this, so don't be alarmed. But we use this information to calculate a covariance matrix and the correlation coefficients, right? And so those are the 2 big takeaways we're going to talk about next, and that I'd like you to try to really think about and have down for your final. So covariance matrix. The sample covariance is a measure of the association between a pair of variables. So if your covariance is 0, it implies that 2 genes or 2 variables are uncorrelated. So you can think of this as age and height in adults, right? After a certain age, just because you're older doesn't mean you're going to be taller. Covariance greater than 0 applies that 2 genes or variables are positively correlated, where covariance less than 0 would imply a negative correlation. So if you need a mental shortcut, you can think of this as slope, right? Slope, some sort of relationship. But an important takeaway is that covariance and variance are different. Variance is what you are most likely more familiar with at this point, and variance is a measure of the spread of data around your regression line, right? So on the left, that tighter coupling of points around the line would be low variance, and then higher variance when there's more scatter around the regression line. Comparatively, covariance is the extent to which corresponding elements from two sets or more of ordered data move in the same direction, right? So if you have y1, y2, y3, so those say there's 3 different variables, you see that the y2, which is the red boxes, and y1, which is the black circles, are positively correlated, right? They both have a positive slope in this case, versus the blue triangles would not be correlated, or at least not strongly correlated with the previous 2 variables. The other important takeaway is this correlation coefficient. So the population correlation is defined to be equal to the covariance, which is what you just calculated, between 2 variables divided by the product of the standard deviation of each variable taken separately. Right? So correlation coefficient is defined as R, which is different from the coefficient of determination, which is R squared, which is what you've been working with up to this point. So in a regression, R squared is the proportion of variance in the dependent variable that is predicted from the independent variable, right? So R squared different than R. So the main takeaways, the rest of the procedure for PCA is a little bit more involved, but I'd be really happy if you could walk away from this lecture knowing, first, what is covariance and how is it different than variance? What is the correlation coefficient R and how is that different from R squared? And why is covariance and R used in PCA instead of variance and R squared? So at the end of this lecture, please go back or pause the lecture now and think about these sort of questions. And if you can't sort of get to what you feel like is a proper understanding, please email me and let's chat more about this sort of difference, and especially why are we using covariance in R and PCA instead of variance in R squared. So circling back around to what PCA tells us, next we're going to look at interpreting the results, sort of which samples cluster together and which variables are correlated to each other and how do we know that. So first, a PCA will give you a result that looks something like this, right? You see how this 2-dimensional space, principal component 1 and principal component 2, and your samples cluster together. Again, this is just sort of mock data, mock results. So if you choose to do a PCA on the sample data, you won't necessarily get something that looks exactly like this. This is just illustrative. But these clusters mean that samples are the most similar, right? So instead of having a bunch of different pairs, a bunch of different XY plots, now you have all that information summarized into one fairly easy to interpret plot. These axes, they're called principal components, are linearly uncorrelated variables, which basically means you can't compare apples to oranges, but if you make them all fruit, it works. So gross oversimplification, but don't worry too much about that part. More importantly, I'd like you to remember that the first principal component, sometimes called factor 1, explains the most variance in the data, where the second principal component, or factor 2, explains the second most variance in the data, third, fourth, and so on and so forth. Normally in results, you'll just see this principal component 1 and principal component 2. But you could keep going if you felt so inclined. And then so you'll see in the results from the paper that I've asked you to read, it says something like 50-some% of the variance is explained by the first 2 principal components. So that's what that means. It's in descending order, principal component 1 having the most, explaining the most variance, and so on. Next, looking at which variables are correlated to each other, there's something called a biplot, which gives you the loading of each variable. And so you won't see a biplot in the paper that I've asked you to read. They're used fairly frequently and also very easy to interpret. So in a biplot, if these 2 vector lines, if they're at 90 degrees, that means that there's no correlation. So genes, in this example, D and C are uncorrelated. So that again would be height and age in adults. If there's an acute angle, that means that two variables are positively correlated. So like genes A and C, for example, versus an obtuse angle, which I realize my face is over the example. Versus an obtuse angle would mean that there's a negative correlation between variables, such as in genes, let's see, F and E. So that is sort of PCA in a nutshell. What I'd like you to take away from it. So next, I'm going to briefly go over the reading assignment. It's still microbial, but it's not genetic necessarily. So please do still read the paper. It's fairly short. I try to pick a short one, promise. But I did want to go over sort of the most important parts of the results to try to focus your reading so you can kind of get the most bang for your buck. So the objectives of the study is Trujillo and Barbosa sampled phytoplankton production and bacterial production for diurnal and seasonal variations, right? So they sampled twice a day, morning and afternoon, and during 2 seasons, in this case, the dry and the rainy season, not winter, summer. And the objective was to estimate phytoplankton production and bacterial production rates along concentrations of carbon, nitrogen, and phosphorus, because those are the sort of the 3 most important building blocks in aquatic biology. And they analyze the data using a PCA and a factorial ANOVA. So a factorial ANOVA is very similar to ANOVAs that you've already worked with in class, except for there's more than one independent variable, so more than one factor. And this all happened in the Rio Doce watershed in Brazil. So first part of the results, principal component analysis was applied with the purpose of reducing the dimensionality, right? So they have lots of different variables and abiotic data, and it searched for gradients in trophic concentration. So the variables they use for their PCA listed to the right, so bacterial and phytoplankton production, total phosphorus, total nitrogen, which are important predictors of phytoplankton growth, dissolved organic carbon as well as dissolved inorganic carbon, and then chlorophyll A, right? So chlorophyll is a photosynthetic pigment, and we use it as a proxy of algal abundance in limnology, or in shorthand, it's CHLA. And the values for these variables are provided in Table 1. Looking at Table 1, you'll see that these are all continuous variables, continuous values, not categorical values. This is an important requirement of PCA. So PCA requires continuous values, but doesn't work as well with categorical data. So you couldn't say, eye and hair color. If you're doing PCA on your classmates, it would have to be something like height or age, right? Continuous, not categorical. Another important thing to notice is that the data is on different scales. So it would have been standardized before PCA was applied. Looking at the results, there's sort of 2 versions of results. One is a comparison using 2 categories. And so this isn't the PCA part, that's just looking at bacterial versus phytoplankton production during the 2 seasons, morning and afternoon at different locations, using this sort of bar chart format. And then the comparison of multiple variables is where they use the PCA. The results, chlorophyll A, total phosphorus, and total nitrogen were positively correlated to the first axes, right? Which makes sense because phosphorus and nitrogen tend to drive algal growth and algal productivity. And dissolved inorganic carbon concentrations were positively correlated to axes 2. In figure 2, this is the first 2 axes account for 58.2% of total variance, right? So that's factor 1, factor 2, or you can give it as principal component 1, principal component 2. The first principal component scores could represent the trophic state of lakes. And then 2 different locations show the highest concentration of chlorophyll A, phosphorus, and nitrogen. They're furthest from the origin of the PCA. So that's basically all I have for the lecture, trying to keep it short and sweet. Some suggested guiding questions for the summaries to get you thinking about for the most important parts of the paper. What is the benefit to using a PCA in this paper? After carefully reading the discussion section, how do the results of the PCA contribute to the objectives of the research? And were you surprised by any of the results? And if so, why? So please read the paper, think about these questions, and then also go back through and think about how correlation and covariance is used in PCA and why is that important. And that's all I have. So hope you guys are all doing well. Please reach out if you need anything inside of class or really outside of class. There's a lot of community resources available too. So if you're having a hard time with the quarantine, we can help sort of point you towards people who can help. But I hope you're all well and staying sane. And that is all I have.